Okay, last class period we finished. We were getting ready to talk about solution phase kinetics, and then begin to compare that with enzyme catalyzed reactions. We stopped somewhere around here talking about a single step process and looking at a reaction coordinate diagram. We had discussed that this was similar to the SN2 reaction that you guys had looked at, as well as some other chemical reactions in organic chemistry, in which you do not have an intermediate. You only have a transition state, okay? So what is the difference between a transition state and an intermediate? What is the major difference?
Well, an intermediate, I don't know how to explain it, but like the intermediate is, the transition state, there's no like solid, I don't know how to explain it, but I know what you mean.
It's really close. I mean,
Like,
I know where you're going.
In the transition state, like, let's say you're doing SN2. Your leaving group comes in as your nucleophile is attacking. It happens at the same time.
Okay.
Transition states do not, or not transition states, intermediates do not.
Okay.
Intermediates are stable molecules on their own.
Yeah.
Transition states can't be isolated.
Okay, that's right. So intermediates are more stable and they can be isolated and proven, right? And transition states are not. They are essentially in transition from reactant to product. Developing catalysts for chemical reactions can either stabilize transition states or they can completely alter the pathway totally. Let's take a look at one that I discussed in Entrochem last semester, and that was the Haber process. Is that the ammonia stuff? Mm-hmm. So the Haber process utilizes nitrogen gas, hydrogen gas to produce ammonia. Well, the catalyst that can be used for this is actually very similar to the catalyst that you find in your catalytic converters on your cars, and these are precious metal catalysts like platinum, iridium, and those types of things. And so what happens is you really create an environment or create a surface by which the two reactants can come into greater proximity to one another and align with one another more readily to reduce the overall energy or activation energy necessary for the reaction to take place. So in here with the transition state, the amount of energy that it takes to get over this hump would be a lot greater if these two gases and these two gaseous molecules had to find each other in the gas phase and to also have proper orientation and sufficient energy for molecular collisions to take place in order to form product. And therefore, you're going to have a greater energy barrier to overcome unless the reaction is catalyzed and you provide this environment. So if you look at the iridium surface, we don't know exactly how this takes place, but we have an idea. That hydrogen binds in a redox fashion to the iridium or to the metal, and again creates this linearity or this environment for the nitrogen to eventually gain hydrogens and to produce the ammonia. So this process is going to happen over and over and over again. So the first go around, you've got this guy, right? And then it's going to do it again, it's going to do it again, it's going to do it again, until eventually it cleaves the nitrogen-nitrogen bonds and gives you 2 molecules of ammonia, okay? But again, without this, you don't have that environment that brings those 2 reactants together in the proper orientation. They have to literally find each other by randomness, right? So this catalyst really greatly enhanced this type of process through changing and actually really stabilizing the environment and how these 2 come together. Again, we don't know exactly how this takes place. One of the reasons why we understand that the hydrogen stays on the metal, and Caleb learned this in his research, thankfully not the hard way, but when you finish using these types of metals for a hydrogenation application, you have to keep them wet. If you leave them around, the hydrogen gas, or the hydrogens that are on them, will snap together and form hydrogen gas, and can do so energetically enough that it will cause a spontaneous fire or an explosion. And so when you're working with a lot of this reagent, then you typically have to keep it wet so that this type of thing doesn't happen. So we talked a little bit about the conditions that are necessary for speeding up a chemical reaction. So how does temperature affect the solution phase reaction kinetics? We all know this. We've done this plenty of times in the lab. You increase the temperature, what does it do to the molecules? That's right, it speeds them up, it makes them faster, you have more molecules that are able to achieve effective conditions. So by increasing the temperature, you're essentially decreasing the overall kinetic energy that's needed to get over that hump. Why can't nature do this same thing?
In our bodies?
Yes. Why can't our bodies use temperature in this same fashion?
We don't work so well whenever our blood is 300 degrees Celsius.
Okay, so what begins to take place?
Denaturization.
Yeah, denaturization. So our bodies have to operate within a specific temperature range in order for us to survive, right? And so this is where catalysts come into play. We can't afford thermodynamically those types of sacrifices. Here is the catalyzed, non-catalyzed reaction coordinate diagram that you guys have seen over and over and over and over again in many classes. Why, or actually, how does a catalyst lower that energy? What are the ways that it can do this? I already showed you one up here. That's true, but there are some similarities. So she's saying the enzymes and catalysts do things differently, but there are some similarities.
Enzymes orient the molecule in a certain direction.
Didn't this orient the molecule in a specific
yes, but I don't think
orientation?
it works in the same way.
Well, this like actually broke down bonds, didn't it?
And
Hold on.
yeah, enzymes wouldn't break bonds.
Ah, so you don't think that there are covalent bonding interactions with enzymes?
Yeah. There are, but I
Okay.
mean, the enzyme itself is not breaking the
Enzymes
bond, right?
like grab the substrate and orient them, physically orient them into their transition.
Yeah.
state.
But isn't that
And then it will cleave and like do the other thing.
So I would say that this is an electronic coordination, right? It's not necessarily grabbing onto it, but there are non-covalent electrostatic interactions, right, that take place, which I would argue that this is similar in that it is an electronic type of stabilization, right? Whereas you would still have an electronic or electrostatic stabilization within an enzyme, right? But what else can a catalyst do? What else can a catalyst do? Enzyme can do the same thing.
So what did we already say it can do?
Yeah, how will it lower that overall kinetic energy to enable that reaction to take place
Well, proteins,
at a faster rate?
I don't know what catalysts, well, I don't know like inorganic catalysts, but proteins have locator domains that know how to track down specific molecules, so it doesn't have to wait till it bumps into it to begin orienting it. It knows how to find it from the get-go.
Yeah, it's highly specific,
Yeah.
right? Okay. What else? Let's go back to this other slide. Okay. Are there chemical reactions in our body that have transition states? So what might an enzyme be able to do?
Stabilize the transitions.
Say again?
Force it into the transitions.
Okay, you're close.
Could it stabilize the transition
So
state?
to stabilize the transition state or to force it to go into a different pathway, okay? So chemists who design catalysts often will study transition states and try to figure out how to manipulate or stabilize those transition states with the catalyst that they're designing. And so that's one similarity between enzymes and catalysts. Or an entirely new pathway can be favored that doesn't even utilize this transition state. And that often happens quite often as well. So this is really transition state theory being applied to enzymatic catalysis. Does anybody have any questions over that reaction coordinate diagram? You guys have seen this plenty of times in other courses.
What does non mean?
Non-catalyzed.
Oh, okay.
Yeah. All right. Take a look at this comparison. Your book makes this comparison of some enzyme catalyzed half-lives and comparing those to the uncatalyzed half-life reaction rates. And it is really alarming. It's pretty amazing to see the change in those half-lives. Look down here at the non-enzymatic. I mean, look how long it would take for that process to happen. I mean, and that's just the half-life, right? One of the emeritus professors at my old university actually would calculate these reaction rate constants and develop computational chemistry to predict some of these, and that's what he did. And I remember him showing, some of these types of uncatalyzed reactions that happen that even organic chemists have come up with, where we've developed catalysts to catalyze a particular reaction that probably would never even take place, and if it did, it would take millions of years for that to actually happen. And it was really interesting to see his perspective on how far we've come in terms of developing catalysts for chemical reactions. Clearly, we wouldn't be able to survive if we had to wait that long for things to happen in our bodies. All right, let's switch gears and look at the entropic and enthopic factors related to transition states. So in this example, there's two reactants that are bound to the sites on the catalyst. And what does this ensure? What does this ensure about these these two reactants.
So again,
What,
I was writing. So.
what is it doing? What is it trying to do? What is the enzyme trying to do? It's trying to ensure that the proper two substrates are catalyzed and that they have, you already discussed it, or you already said it, Heaven, the proper orientation, right, for those two to come together. Because again, if you don't have the proximity and you don't have the proper orientation, then the overall entropy is not going to be favorable for this to happen, okay? So you're talking about forcing the delta S of the transition state to literally be less negative in that process. Any questions on that? So what are the two different models for enzyme substrate binding?
There's the bad one, it's the lock and key method.
There's the bad one, the lock and key method, okay? Actually, I want to come back to this slide in just a minute. So, which one is which? Here, I'll throw this up here. And remember those things that kids look at and they're like, what is the same and what is different about? So take a look. What's the same and what is different?
That one changes shape.
Which one?
The red one.
The one on the right. Where does it change shape at?
After the substrate is binded to it.
After the substrate has bound to the enzyme
The binding site,
at the binding site. So which one's the lock and key method, Caleb?
the left one.
The one on the left. Imagine if you had a key and you put it into the lock, and every time you put it into the lock changed the key. Just a little bit. It changed the key just a little bit, right? That's essentially what the induced fit model is referring to, okay? And that is that when the enzyme and the substrate bind together, that actually both are changed a little bit, okay? The lock and the key together. And so in terms of like real specificity, we thought this before was a very rigid type of a system. Drug design and designing molecules for the induced fit model actually used to be more rigid than it actually is today. We actually had thought that it was important to have these molecules fit into the cleft of the enzyme or the protein in a very rigid manner, very specific to that. And now we've determined, and this has of course been through experimental data, that there should be some flexibility to that in order for greater binding to actually be achieved. Caleb, you were gonna
I was gonna say, is that kind of like the fluid mosaic model, but like with these now? It's not cell membrane anymore, it's smaller, but it's kind of the same idea at least.
explain what you're thinking.
Well, like in the fluid mosaic, we know that the cell membrane is not a rigid structure. It moves and gives and bends ever so slightly. And so it's kind of the same thing with this, because it does have a defined shape, but it can, whenever something applies pressure to it, or it needs to because of its environment, it will change shape to alleviate stress, which I think is
Abigail, did you want to add to that?
No, I was just being an idiot and was staring at the green pieces.
She
not the
leans
others.
over, she goes,
For the inside.
where did it change?
Oh, okay.
I'm a hardcore.
It's actually funny because, when you do look at stuff like that, it's just like the kids, right? The kids are looking at those and like, all right, what's different and what's the same? And your perspective, it's all about your perspective, right? I think when I first looked at this, it was like, okay, yeah, what has changed? The middles are round, and then I'm like, oh yeah, there's the square and then the round.
The middle is around on both of them.
honest, I
It's
think
true.
more adults play those games than kids.
Oh, you think so?
Don't call me
That
out.
might be the
I
case.
still like them. I enjoy them.
Believe me, if I crossword puzzles are
So,
Gonna fail the DNA work, obviously, the DNA.
let's go back. Let's go back. So, enzymes can alter reaction pathways. Catalysts can do this too, right? Catalysts can alter reaction pathways, and oftentimes they do, taking the reaction on it in a completely different direction, lowering that overall activation energy necessary to form product. What is this? What is this graph showing you? What is this reaction coordinate diagram showing you?
There's two transition states.
No.
I'll show you a small snippet of an enzyme.
Okay. What else?
And show you the energy difference between a transition state and an intermediate state.
Sure. Okay.
What is the enzyme making there be two reactions because you have G1 and G2?
So you were right in the fact that there's two transition states in the intermediate pathway, right? And that's true, there's two humps, okay? What this is trying to refer to, or trying to tell you, that an enzyme can take a to B molecule or molecular reaction, take the transition state and either stabilize it or create an entirely new pathway that actually has a more stable intermediate and allow the reaction to proceed at lower energy simply based upon that transition, or simply based upon that activation energy to get to the intermediate, okay?
Functional intermediate.
Maybe. What I'm saying is that even though there are two steps in the next process, right, the catalyzed process, which is the red one, that is still more favorable than having a transition state that's not stabilized. That's what I'm getting at. Did I lose you?
Does this happen in like live organisms or?
Yes.
is it-
Yes, yes.
How does it know when to do the adapters?
This is a hypothetical scenario where if that reaction happened in your body, okay, if it happened on the time scale that you needed it to happen, because that might not be the case looking back, that the enzyme is so well designed and so specific that it has figured out that the intermediate and the stability of the intermediate, the amount of energy that it takes to reach that intermediate, is so much more favorable than having to go through a transition state that a normal or typical chemical reaction would happen in your body with those same molecules, essentially. Okay? And I don't know how oftentimes this happens, right? I think, again, it just depends on the system. I mean, we have so many enzymes, we don't know what they all do, right? We haven't even discovered all of them. Any other questions on this? Okay, so an enzyme can alter the reaction pathway, and it can create one that includes one or more intermediates that may resemble the transition state, but it's lower energy than the transition state. Let's take a look at this induced fit model study for hexakinase. So hexakinase is an enzyme that catalyzes glucose to glucose 6-phosphate at the beginning of the of glycolysis. This happens in metabolizing or metabolic pathways in our body. And let's take a look at the three-dimensional conformations. Okay, so before glucose binds, you've got the cleft in which glucose is going to bind within there. There's actually a metal that is, I think this is magnesium 2+. Is it on the previous slide? It is not. It is though. Magnesium 2+ is involved in this process, and there are a lot of metal-mediated catalysis reactions that actually involve enzymes in our body. Metals are really important for enzyme catalysis, and I think, what does your book say? I thought that it said like there's a certain percentage of all the enzymes that's been discovered in the human body. There's a certain percentage that are actually metal-mediated processes. Let's see if I can find that number. One third. Over 1/3 of enzymes characterized to date contain metal ions in their activation sites or in their active sites, and these are called Metalloenzymes. So there's a whole separate group of biochemists that study metalloenzymes and characterize metalloenzymes. Okay. So before the glucose binds, that's what it looks like. And after glucose binding, that's what it looks like. What's happened? Take a look at it.
It went Pac-Man.
Let's go to the next slide and let's look at the next two slides and let's kind of go back and forth and look at this. So there's before, and there's after.
You've had a shape change over here. Oh, actually, the whole thing changed. Shape. Looks like it curls more over.
Caleb's right. It does look like it does kind of a Pac-Man, right? What's really cool is that, I mean, that proves the induced fit model right there, right? That's evidence that this exact type of situation takes place. So not only does the protein or the enzyme change shape, okay, but there are actually slight conformational changes that actually happen with substrates too, okay? You think about like strain. You know, when we were in organic chemistry and we were looking at boat and chair conformations, right? And you know that the boat conformation has to go through a half chair before it reaches the full chair conformation. Your book actually talks about this, okay? The conformational changes that can occur even in sugars are the same. You can get these types of changes in ring conformations, and the enzyme should be able to stabilize some of those conformations in order for chemical reactions to take place at proper orientation and proper angles. And so again, it's not just the enzyme that's actually changing the shape, but it's the key too. It's that substrate as well that can be stabilized in a different orientation. So here's a reaction coordinate diagram. You can see that it looks very different for a simple enzyme catalyzed reaction. What do you notice? Right offhand. So the blue curve is the free energy profile for a non-catalyzed reaction.
The enzyme substrate complex is way lower than some of the transition state.
Yeah, but I mean, even look at the activation energy needed to achieve that first complex, right?
Needs less energy to activate, or no, there's a small hump right there.
Yeah, there's a small hump there, but that well is, I mean, it's big actually, right? So you have that initial well of stability involved, then you've got more activation energy, right, that needs to take place. That is the enzyme right there, bound to the substrate, already putting it into the proper orientation that it needs to make the chemical changes that it needs to make in order to overcome that next hump, right? So what happens after the next hump?
You have your enzyme and product.
You now have your enzyme, it's still bound to the substrate, but now it's already made those chemical changes to the molecule that's necessary. That previous slide would have been what? What would have been the change?
The chomp,
The chomp and then,
And
the
the-
addition of the phosphate.
yeah, and then the phosphorylation, okay? So clearly ATP has to be involved, right, in that change, because if ATP is just floating around and it's not right there, is it going to take place in that? Is it going to be energetically favorable for that reaction to take place?
For it to break away.
It's not. So that means that it has to be bound with it, right? It's gotta be right there in proximity in order for that phosphate to transfer. Yep, okay. Fancy slide. Okay. Once the product is completed, then the enzyme releases the product. And does the enzyme change?
After it releases, it changes shape.
Well, it would go back to the
During
conformation it
reaction,
had
it goes
before
back.
it binded, wouldn't it?
Doesn't it make sense that it would have to be a reversible change?
Yeah, it's gonna be a reusable.
Because
enzyme.
If it's gonna have to be reused, absolutely. So even though the enzyme changes in its conformation during the process, it has to maintain its overall shape in order for that reaction to take place all over again. Let's take a look at an enzyme catalyzed ester cleavage. This is in your book as well. You guys have not really seen much carbonyl chemistry. That really is the last chapter I wanted to cover in organic chemistry. I hope we can get through some of that within the next week or two. But take a look at the transition state. The transition state for this is a tetrahedral oxyanion type transition state whereby the enzyme has a hydroxy group that then attacks the ester carbonyl because that is polar, it's polarized, right? And then you're looking at trying to stabilize that transition state by a couple different means. Either an electrostatic stabilization by various charged groups on amino acid residues. Take a look. That would be the N-terminus, right? Stabilizing that charge. And then the C-terminus where you've got the carboxylate stabilizing this charge. What's the other one? It looks like hydrogen bonding.
Is it just like the proximity of the charges that stabilizes it?
Okay,
It looks like the resonance occurred and one oxygen got a gamma carbonyl, one has an extra pair and it actually bonds to the hydrogen, but it's still bound to the other ones.
okay. So this has to do with proton transfer, and this is general acid-base catalysis, or GABC. Your book has a mnemonic for that. So GABC, and with that, anybody know that residue? That amino acid residue?
Wait, which one are you looking
It is histidine. And as a matter of fact, histidine is very common for GABC catalysis. And that is due to its ability to accept or donate protons at physiological pH. And so that you actually see that quite often. Good job. Nice work.
What are those lines? The red lines on there.
So the red lines on there, that's why she was saying it looked like hydrogen bonding, okay? They're just showing you where, I think where that's taking place, where that proton transfer can take place. Yeah.
So it can take place on eight and also on the same time.
I would say that it could probably do it on both. Yeah, absolutely. I'm actually not that familiar with GABC catalysis. We can
It looks like an enzyme.
we can read a little bit more about that, and I can maybe better answer that later. Yep. Your book has this lysozyme case study in it. I'd like for you to read this case study, and we'll go over it next class period. I also have another case study that's not in your book, an enzyme case study that I would like for you guys to do as a group activity, probably on Friday. We'll do that, and we'll come back on Wednesday and talk about this particular case study and look at the mechanism, and you guys can discuss that in groups as well. Okay? All right, you guys have a good day.